Hello students, in today's class we are going to learn about membrane transport and specifically discuss about active and passive transport. So here is the diagrammatic representation of different types of transport mechanisms. As you can see, active transport mechanism is the one in which there is energy input in the form of ATP. Passive transport is divided into different types such as simple diffusion and facilitated diffusion. Facilitated diffusion is again based on two types, that is the transport that is mediated through channel proteins and through carrier proteins. So we are going to look into more details of each of these different types of transport. So passive transport is the simplest mechanism by which molecules can cross across the plasma membrane. It is also known as passive diffusion. Now, during passive diffusion, a molecule will simply dissolve in the phospholipid bilayer and will diffuse across the other side of the membrane. So here no membrane proteins are involved and the direction of the transport is basically determined by the concentration gradient. So if the concentration is higher on the outside, the net flow of molecule will take place towards the inner side. So only small molecules and relatively hydrophobic molecules are able to diffuse through the phospholipid layer. Examples include gases such as oxygen and carbon dioxide, hydrophobic molecules such as benzene, and small polar but uncharged molecules such as water and ethanol can diffuse across the plasma membrane. So here we have a diagrammatic representation of what type of molecules can be permeable across the phospholipid bilayer and which molecules cannot permeate directly by diffusion across the phospholipid bilayer. So molecules including gases such as oxygen and carbon dioxide, hydrophobic molecules such as benzene, and small polar molecules such as water and ethanol, they can easily diffuse across the lipid bilayer. Other molecules, including amino acids, which are categorized as large polar molecules, and charged ions such as sodium ions, calcium ions, etc., are unable to diffuse across the lipid bilayer. Next is facilitated diffusion and carrier proteins. So facilitated diffusion is like passive diffusion, in which it involves the movement of molecules determined by the concentration gradient. That is, the movement of molecules will take place from the high concentration to the low concentration location. Here, no external input of energy is required in the form of ATP or in the form of electric potential, etc. However, facilitated diffusion also differs from passive diffusion in that it requires certain transporter molecules, and these molecules basically are not able to dissolve and transport themselves across the phospholipid bilayer, and instead these molecules will make use of certain passages that are made up of protein molecules. These allow the transport of specific molecules across the membrane in such a way that these molecules can avoid interaction with the hydrophobic lipid bilayer. Facilitated diffusion therefore allows polar and charged molecules such as carbohydrates, amino acid, nucleosides, ions to cross across the plasma membrane. Two classes of proteins that mediate facilitated diffusion are recognized. That is the carrier proteins and the channel proteins. So the carrier proteins are those proteins that bind specific molecules to be transported on one side of the membrane, and then they undergo conformational change that will allow the molecule to pass through the cell membrane to the other side. On the other hand, channel proteins, they form open pores through which the membrane will allow free diffusion of any molecules of appropriate size and charge. So the carrier proteins are responsible for facilitated diffusion of sugar. So we can look at a specific example of how the uptake of glucose molecule can take place with the help of carrier protein. So here is a diagrammatic representation of the model for facilitated diffusion of glucose molecule with the help of glucose carrier protein. So the glucose transporter, it functions by alternating in two conformations. In the first conformation, it allows binding of glucose, and it is facing in such a way such that the glucose binding can take place to from the extracellular side of the cell membrane. As soon as the molecule glucose binding will take place to the transporter molecule, the transporter undergoes a conformational change such that it opens towards the cytosolic phase of the plasma membrane, and this will allow entry of the glucose molecule into the cell. This transport of glucose molecule is along the concentration gradient that is happening from the location of high concentration to the location of low concentration. So as soon as the glucose molecule will enter, it dissociates from the transporter, and the transporter will return to its original conformation and is now available for binding a fresh glucose molecule. The second type of molecules that will allow transport of uh, different molecules includes the channel proteins. And the channel proteins, they simply form open pores in the membrane, allowing small molecules of appropriate size and charge to pass freely through the lipid bilayer. One group of channel proteins is porins, which will permit free passage of ions and small polar molecules through the outer membrane of bacteria. So here we have a representation of a model of ion channel, which will be present in two conformation. So these type of ion channels are not always in an open state. They basically are gated channels, and the gate can be present in two conformation, either closed conformation or open conformation. So when it is in closed conformation, it will not allow passage of molecules, but when it is in open conformation, it will allow free passage of molecules of appropriate size and charge through the ion channels. So coming to the properties of ion channels that are important for their function. Firstly, the transport to the channels is extremely rapid if you compare the transport through the carrier or transporters. The transport through the channels is extremely fast. More than 1 million molecules per second will flow through the open channels, and the flow rate is 1,000 times greater than the transport by the carrier proteins. As we had just seen in case of glucose carrier proteins, it will transport one molecule of glucose at a time. So it is a relatively slow process. Secondly, ion channels are highly selective because the narrow pores in the channel restrict passage to ions of appropriate size and charge only. So, specific channel proteins will allow passage of ions such as sodium, potassium, calcium, chlorine across the membrane. Thirdly, most ion channels are not permanently open. Instead, the opening of the channel is regulated by the gates, which will transiently open in response to the specific stimuli. Some channels are called as ligand gated channels that will open the gates by a stimulus that is binding of a neurotransmitters or other signaling molecules. Other type of channels are basically voltage gated channels that will open in response to the changes in the electric potential across the membrane. Next, we will discuss about active transport. So how does it differ from the passive transport? So in passive transport, the transported molecules always move down its electrochemical gradient and is not accumulated above the equilibrium concentration. On the contrary, in active transport, you can have transport or accumulation of solute against the concentration gradient. That is, the transport can occur from a location of its low concentration to the location of its high concentration. So it can move in the opposite direction of the concentration gradient. And obviously, because of that, it requires energy in the form of ATP. So we can either have a ATP driven pump that is involved, that is going to transport the molecule, or in certain bacteria there are light driven pumps, for example in photosynthetic organisms, that will provide the required energy for transport of molecules against the concentration gradient. Again, active transport can be categorized into primary active transport and secondary active transport. So as you can see diagrammatically, in primary active transport, you are having direct transport of one metabolite. For example, metabolite X here is transported from the cell to the outside of the cell against the concentration gradient. It is an energy requiring process in the form of ATP, which is converted into ADP, and the energy release is used for the transport of the molecule. Now, on the contrary, in secondary active transport, what we have is, first we have transport of one metabolite. For example, the metabolite X is again transported across the membrane against the concentration gradient by making use of energy. For example, here we have ATP hydrolysis, and that leads to accumulation of one of the metabolite in higher concentration across the membrane. So you'll have more concentration of metabolite X on the outside than compared to the inner side. Now, this is basically an electrochemical gradient, and the transport of metabolite X from the outside to the inside of the cell again is energetically favorable reaction, since it is moving according to the concentration gradient. Now we have certain transporters. In secondary active transport, what we have is, we have these transporters that will transport the metabolite X across the membrane, or across the transporter back to the cell cytosol, and during the transport it will simultaneously also allow other molecule to be transported inside the cell. So since in secondary active transport, first we have the transport of one metabolite that is going to create a electrochemical gradient or a gradient that is going to, that gradient that is generated will be utilized for transporting the second metabolite across the membrane. This type of transport is known as secondary active transport. So the active transport can be driven by ion gradients, and three different types of transporters or ion gradients are recognized. So uniporters are those type of transporters that can carry one molecule or one ion in one particular direction. Then we have symporters that will carry two different molecules or ion both in the same direction. Okay, so these are also known as co-transporters. Then we also have antiporters or exchangers that carries two different molecules or ions but in opposite directions. So these type of transport systems can be present both in case of active as well as passive transport. So here we have an example of active transport of glucose that is driven by another molecule, that is sodium plus ions. And this example is commonly seen in the epithelial cells that line the intestine, where the intestinal glucose molecules are to be picked up into the cell, and this is against their concentration gradient. So as you can see, first sodium plus ions are transported across the membrane through whatever mechanism. It can be active transport, and as a result you can see there is more accumulation of sodium plus ions on the outside compared to the inside of the cell, and this will facilitate the transport of the second molecule. So the transporter will, what it will do is it will bind the sodium 2 plus ions and will also allow binding of glucose molecule. So glucose molecule here, after binding to the transporter, the orientation of the transporter will change and will open towards the inside, and you will find that the sodium plus ions as well as the glucose will enter inside the cell. Once the molecules will leave the transporter, it will again come to the original conformation. So this is an example of a symporter where the glucose ions are moving against their concentration gradient, that is from low concentration to high concentration across the cell membrane, and this is facilitated by the presence of high concentration of sodium ions in the outside of the cell. The same points are mentioned in this slide for your reference. And that's it about active and passive transport. Thank you very much, and kindly like, share, and subscribe if you like the content.